You know how CAR T cell therapy has just been this incredible, like, game changer for cancer treatment? Such amazing hope there.
Absolutely phenomenal progress.
But the big puzzle, the thing we're always trying to crack, is how do we make those amazing responses last? How do we get those CAR T cells to really stick around long term and keep the cancer gone for good?
Right, the persistence problem. It's key.
Exactly. So this deep dive, we're jumping right into some really fascinating new research that tackles that specific challenge head on, getting that long-term effectiveness, or, well, what scientists call persistence.
Mm-hmm. And our main source today is this really detailed research paper. It digs deep into how these engineered Keysight T cells actually, decide their fate long term.
Yeah. Do they become those, like, fast-acting effector cells? The ones that go in for the immediate kill?
Or do they shift into being these long-lived memory cells, the ones that hang around for surveillance, basically?
So our mission, really, for this deep dive is to follow the researchers' thinking. Like, step by step, what did they see first that made them go, What questions did they ask? And how did they, set up their experiments to peel back the layers on this? Because they weren't starting completely blind, right? They had this idea from our own natural T cells.
Yeah.
Something called asymmetric cell division.
That's right. It was the jumping off point. See, for a while, scientists knew that when a normal T cell meets a threat, like a virus, it divides. But sometimes the two daughter cells aren't identical twins.
Oh, interesting.
Yeah, one might be, primed for action right away, the effector type, while the other is more set up to remember the threat for the future, the memory type. So these researchers thought, well, could that be happening with C-cell cells too?
Okay.
And if it is, could this be like a really crucial piece for making them work better long-term against cancer?
And it's such a critical question because, the paper really points this out. CRT therapy works wonders, sometimes achieving complete remission.
It can be amazing.
Sadly, a pretty significant number of patients still relapse later on. And often, that relapse seems linked to the CLC cell tubes just not sticking around.
They don't persist.
Yeah, they do their job up front fantastically, but then they kind of, fade out. So figuring out how some become these long-term memory guards while others don't is, it's a really high-stakes problem.
Absolutely. The researchers knew there was this link. It was pretty well known.
Mm-hmm.
If you see a good, strong expansion of CAR T cells early on, right after infusion, that often means better long-term results.
Okay.
But the how was the mystery. How do they actually split into these short-lived effectors versus the long-lived memory types?
Yeah.
Especially in human CAR T cells, the nuts and bolts were pretty unclear.
Right.
And this whole idea of asymmetric division, whether that played a role like in natural T cells, that was a complete unknown for CAR T cells.
Okay, so they've got this clue from normal T cells and this really urgent clinical need. What was their breakthrough? How did they even start to tell these two daughter cells apart when a CAR T cell first divides after seeing a cancer cell?
Right. This is where they got really clever. They took a technique called lip-estic. Bit of a mouthful. It stands for labeling immune partnerships by SORT-tagging intercellular contacts, and they adapted it.
Okay, lip-estic.
Now, normally you might track T cells using markers like CD8, right? But CAR T cells, they're different. They don't recognize cancer the same way normal T cells recognize viruses, using that whole MHC presentation system.
Right. The CIR itself is engineered to grab onto a specific target directly on the cancer cell.
Mm-hmm.
It bypasses the usual process.
Exactly. So the researchers thought, okay, let's focus on the CAR molecule itself, the engineered part. The core idea behind their tweaked lip-estic was to use that direct CAR-target interaction to label the daughter cells right at that first division.
Huh.
This let them really, for the first time, properly separate and study the immediate offspring of a CAR T cell right after it meets the cancer, which is, totally crucial for understanding their long-term fate.
Okay, so break that down for me. How do you actually label something during cell division based on contact? That sounds complicated.
It is pretty neat. So imagine the CAR T cell latches onto the cancer cell. They use this enzyme, sortis A, and they tweaked the CIR molecule itself to have a little tag. Then they loaded the target cancer cell with this biotinylated peptide. Think of it like a tiny sticky note with a chemical handle. Biotin.
Okay, a sticky note on the cancer cell.
Right. So when the CAR grabs the target, the sortis A enzyme, which is sitting there on the cancer cell surface, acts like molecular scissors and glue. It snips off that biotin sticky note and attaches it right onto the CIR molecule of the T cell.
So only the T cells that are actually touching a target cell get this sticky note stuck onto their CIR.
Precisely. Then, when that CAR T cell divides for the first time, that label, that biotin peptide, it gets passed down to only one of the two daughter cells.
Only one. How?
Well, the one that was physically closest to, or maybe still in contact with, the target cell during that labeling moment, they called this one the proximal daughter, close proximity, right?
Okay, proximal.
The other daughter cell, the one further away or not directly touching at that instant, doesn't get the label. That's the distal daughter.
That is incredibly smart. So now they have a definitive way to sort these two populations labeled proximal, unlabeled distal. And they actually watched this happen with imaging.
You did. Live cell imaging. They could literally watch these CAR T cells divide under the microscope after labeling. And clear as day, you see the fluorescent tag, which was linked to the biotin, ending up in just one of the daughter cells. The other one stays dark.
Wow.
It was, direct visual proof of this asymmetric thing happening, at least with the labeled CARs.
Okay, powerful tool in hand. Proximal versus distal. What did they find out about them right after that first split? Were they different?
Oh, yeah. Right away, some really interesting, distinct features popped out. The proximal daughters, the labeled ones, they tended to be physically bigger.
Bigger, okay.
And they had more CD25 on their surface. That's a classic marker for T cell activation. Means they're more, fired up. And critically, these proximal cells started multiplying, proliferating much faster in those first few days.
Sounds like the cell that's ready for immediate action, right?
Exactly. And then they looked at metabolism. How are these cells generating energy? More differences. The proximal daughters had really cranked up their metabolism. They were heavily reliant on glycolysis.
Which is the fast energy pathway.
Right. It's quick, good for rapid growth, but maybe not as sustainable long term. It fits that profile of an effector cell gearing up.
And the distal ones, the unlabeled daughters.
Much more subdued metabolically, only a slight increase compared to resting cells, and they seem to lean more towards oxidative phosphorylation.
The slower, more efficient pathway.
Yeah, often linked with cells that are more in a resting state, or maybe heading towards memory. So even these very early metabolic snapshots strongly hinted that this asymmetric division was kicking off two very different functional programs.
Fascinating. But SARG T cells, they aren't all the same, are they?
Yeah.
You've got CD4, CD8s, and they can come from different starting T cells.
Yeah.
Did this asymmetric division happen across the board?
Crucial question. And yeah, they looked into that to see how general this was. They found this pattern, the differences in size, CD25, proliferation. It wasn't just in CD8 CAR Ts. They saw a similar thing in CD4 CRT cells too.
Okay.
And it held up even when they changed the ratio of CAR T cells to target cells in the dish. They even saw hints of this asymmetric labeling happening in vivo, in animals.
What about where the CAR T cells originally came from? Like if you make them from naive T cells versus already activated effector T cells?
Another good point they checked, CRT cells made from naive T cells. They showed this clear asymmetry in the first division. Now, interestingly, CAR-Ts made from effector cells still showed the trend in proliferation differences proximal faster than distal, but the size and CD25 differences weren't quite as stark.
So still a difference, but maybe less pronounced if they started as effectors.
Mm-hmm.
Seems like this asymmetric division is pretty common in CAR Ts then. Yeah. But was it just a one-shot deal? Could those distal cells, the unlabeled ones, do it again if they saw another cancer cell?
Exactly what they wanted to know. So they did this really neat follow-up. They sorted out the first generation proximal and distal cells using the lip-uvestic label. Then they showed them target cells again, but this time using a different colored lip-uvestic label.
Oh, okay. Round 2.
And what they found was really telling. Those distal daughter cells, they could absolutely undergo asymmetric division again on that second encounter. They produced daughters with different proliferation rates, different markers.
Wow.
It suggested that this potential to differentiate, it wasn't just a one-time thing, it was something those distal cells carried forward.
That's really key. It means those distal cells aren't just some static intermediate. They can keep generating diversity when they re-encounter the tumor.
Okay, so initial differences observed, potential for repeated asymmetry. The million-dollar question, did they actually behave differently long-term? Did one group actually lead to better cancer control?
Yep. Time for the in vivo test. Mouse models. They sorted the proximal and distal daughters, injected these separate groups into different mice, let them settle in a bit, then challenged the mice with leukemia cells.
Okay, the real test.
And the results, pretty dramatic. The mice that got the distal daughter cells showed way better persistence of the SOAR T cells over time.
Higher numbers.
Yeah, higher counts in the blood. And most importantly, those mice were much, much better at controlling the leukemia growth. The mice that got the proximal daughters, Not nearly the same level of long-term tumor control.
Wow, that's about as clear as it gets, functionally speaking. And didn't the paper say something about the distal ones staying more naive-like in the mice? Yes, exactly. When they looked at the CAR T cells in the spleens later on, the cells that came from those original distal daughters had a higher proportion of cells with naive T cell markers. It suggests they had this better capacity for self-renewal in vivo.
Which is what you want from a memory cell, right? The ability to keep making copies of itself.
Precisely, a hallmark of long-lived memory.
Okay, so distal, persistence and control. But then there was this weird twist, wasn't there? Didn't they find that these supposedly memory-bound distal cells could actually kill cancer cells really well, at least initially? Seemed kind of backwards.
It does seem counterintuitive, yeah. It was definitely an unexpected finding, but super interesting. They saw that in the first few days, like day 1 to 3 after division, those distal daughters were just as good at killing target cells. Their cytolytic activity was comparable to the proximal daughters.
The ones that look like the dedicated killers.
Right, but here's the key. In the distal daughters, that killing ability was transient. It dropped off after about day 4, whereas the proximal daughters kept up their high killing rate, at least for that early period.
Huh. So it's like the distal cells have this little burst of effector power right at the start, before they fully commit to the long-term memory game.
That seems to be the picture.
That's really interesting. It might even help explain some debates in immunology about whether memory cells come straight from naive cells, or if they pass through an effector stage first. Maybe they do have a brief effector-like moment.
That's exactly the implication the researchers raise. It hints at this cellular plasticity. Cells heading for memory aren't necessarily just sitting quietly from the start. So to really get under the hood and see what was driving these different behaviors, they went deep with single-cell analysis.
Right, the fancy stuff. Multi-omics, looking at everything at once in single cells. What did that show?
Yeah, they threw the book at it, looked at surface proteins, RNA sequencing for gene activity, even T-cell receptor clonality to track cell lineages, and the surface protein data. It just painted a really clear picture. Proximal and distal daughters were distinctly separate populations.
How so?
Well, the overall protein look of the proximal daughters was very similar to activated T cells that hadn't divided yet. The distal daughters, though, their surface proteome looked much more like resting T cells.
Any specific proteins that stood out?
Yeah, confirming their earlier flow data, distal daughters had more CD45RA and CD5. Proximal had more CD25, the activation marker. But one really surprising thing was that distal daughters had more of the natural T-cell receptor and the CD8 co-receptor on their surface.
Oh, that's different from normal T-cell division.
Apparently so. It seems to be a unique feature of this CAR T-cell asymmetric division.
Interesting. And they saw bits of the cancer cells stuck to the proximal ones, too.
That's right. Things like CD19, CD10. The CAR T cells weren't making these proteins, so the thinking is it's trogocytosis. The T cells literally nibbling bits off the target cells. Another sign of that intense interaction. So yeah, the surface protein story really backed up this idea of rapid divergence right from Division 1.
Okay, surface looks different. What about inside, the genes? What was the RNA sequencing telling them?
Huge differences there, too. Even if you took cells that looked similar on the outside based on markers, say, both looked naive-like, if one was proximal and one was distal, their internal gene expression programs were globally different.
Wow.
Specifically, those naive-like distal daughters had higher levels of RNA for genes we know are crucial for memory and survival, things like LEF1, TCF7, CCR7, IL7R, KLF2. Classic memory players.
Okay.
And gene set analysis just confirmed it. Distal cells lit up naive gene signatures, proximal cells lit up effector, and metabolism signatures.
So the genes were telling the exact same story as the function and metabolism. And they used that RNA velocity thing, too, to see if it was inherited RNA or new gene activity.
Exactly. RNA velocity gives you a sense of direction. Are gene levels actively going up or down? It helped them figure out if the differences were just because RNA got split unevenly during division, or if the cells were actively turning genes on or off after dividing.
And the answer was?
Kind of both, actually. For some key memory genes in distal cells, like IL7R and KLF2, they saw evidence they were being actively switched on after division. For others, like CCR7, it looked more like they just inherited more of that RNA to begin with.
So it's a mix of inheritance and active programming.
Right. It really showed how this asymmetry gets set up through multiple layers. And when they mapped out the likely developmental paths based on gene expression, the trajectories, they saw the proximal and distal daughters heading in basically opposite directions. Complete divergence from that very first split.
Like two trains leaving the station on different tracks from the start. So to figure out who was driving these different gene programs, they looked at the master switches, the transcription factors.
Precisely. They use computational tools to infer which transcription factors, those proteins that control gene networks or regulins, were most active in each population.
And did clear patterns emerge?
Absolutely.
Yeah.
In the proximal daughters, you saw higher activity of factors linked with, well, things you'd expect. Apoptosis or programmed cell death, really fast proliferation, pushing towards that effector fate. Names like TP73, E2F factors, MYBL1, YBX1 make sense for a short-lived killer cell.
Okay. And the distal daughters?
The opposite, really. Increased activity of factors known to put the brakes on proliferation, promote self-renewal, lock in that memory state. You see names like KLF2, IRF family members, STAT1, KDM5B, REST, FLI1, MXT4, and importantly, IKZF1.
IKZF1, Icaros, right. So one set of factors hits the gas towards the effector death, the other hits the brakes and steers towards memory survival. And they actually tested IKZF1 directly.
They did. Crucial validation step. They used CRISPR gene editing to knock out IKZF1 specifically in those distal daughter cells.
Right.
Remember, IKZF1 is known to kind of restrain T cells, promote memory.
Right, so what happened when they deleted it?
The distal daughters started to lose their memory identity. They lost CD45RA expression, started looking more like the proximal daughters phenotypically and functionally.
Let me guess, they didn't persist as well.
Exactly. When they put these IKZF1 knockout distal cells into the mice, they didn't persist nearly as well, and they couldn't control the tumor growth as effectively as normal distal cells.
Wow. So that really nails IKZF1 as a key player in making those distal cells behave like long-term memory cells.
It provides really strong functional evidence. Yeah, it shows these molecular differences aren't just correlations. They're actively driven by these transcription factor networks.
Okay, so let's pull all this amazing work together. What are the big takeaways here? What does this mean for a CAR T therapy, and maybe even beyond?
Well, first, it pretty convincingly establishes asymmetric cell division as a core mechanism for how human memory CAR T cells are actually made. It shows that right from division 1, you get these two distinct daughters, molecularly different, heading down different paths.
One for now, one for later.
Pretty much. And that finding about the distal daughters having that brief effector phase, that's actually quite important. As we said, it might help smooth over some old debates about how memory T cells even arise.
Right.
And therapeutically, this is maybe the most exciting part. Identifying key regulators like IKZF1 gives us potential handle, actual molecular targets, if we can figure out how to, say, nudge the balance during that first division.
Tip the scales towards making more distal memory type cells.
Exactly. If we could encourage the formation of more of those long-lived memory precursors right from the start, that could potentially lead to much more durable, longer-lasting responses for patients. That's the hope.
So for everyone listening, the really big idea to grasp is this. iKT cells, when they first divide after seeing cancer, aren't just making clones. It's this incredibly elegant split, creating two specialized cell types. One's built for the immediate fight, the other's built to last, to keep watch. It's a fundamental built-in strategy we're just now understanding deeply at the molecular level.
Absolutely, and the significance is huge. This work gives us a whole new way to think about CAR T cell differentiation, and more importantly, it points towards concrete ways we might be able to engineer these cells to be even better, even more persistent killers of cancer. It's about getting in early and influencing that fundamental fate decision.
It really makes you think, doesn't it? If we can understand and maybe even control this asymmetric split for CAR T cells, what else could this apply to? Where else in medicine do we need long-term immune responses?
Vaccines, for instance, autoimmune diseases.
Right. Could we use this knowledge to make vaccines that give more durable immunity? Or maybe an autoimmune disease, could we tweak things to discourage certain long-lived problem cells? The idea of being able to steer cell fate that early on, well, that's a really powerful concept to chew on.